Hello everyone, welcome back to KaushalBioPrep. In today's video lecture, we are going to talk about animal biotechnology. Basically, we are going to explore the area under gene manipulation. So, animal biotechnology is a combination of science and engineering. So, there is bio and technology, right? So, we are using engineering and the biological world, and it is basically useful for transforming the living organism. So, the main objective of this particular field is to make the products and to improve the animals or to develop microorganism for specific agriculture uses. So these are only few objectives of the animal biotechnology that I have mentioned here, but there are several others, right? So one by one we are going to discuss what actually are the purpose of animal biotechnology. So basically you must have heard about the transgenic animals. So those are the products of the animal biotechnology. This is the best example of animal biotechnology. And you may have heard about the genetically engineering crops, right? So basically they are called genetically modified organism and GMO crops. So they have been in existence since a very long time, and they are also the product of the animal biotechnology. So what is biotechnology basically? So biotechnology is the application of the biological knowledge and the techniques, and they both are merged to create products and improve the processes and solve problems in various sectors such as health, agriculture, industry and environment. So these are the basic sectors in which biotechnology works, and basically it enhances the productivity of these sectors, and it enhances the cellular and biomolecular processes for technological advancement to benefit the societal sustainability. So basically we are using it for the benefit. So whatever the product is there on which we are working along with the biotechnology, we will be enhancing that product or increasing the productivity. So it combines the biology with engineering, genetics, chemistry and computer science to manipulate the living system or any part of that system. So biotechnology's scope is vast, continuously expanding as the science expands. So it covers multiple interdisciplinary branches and applications. So first of all, we'll study about the medical biotechnology, which is also called the red biotechnology. So under this, there is development of the recombinant pharmaceuticals like insulin and vaccines are being developed under this technology. So and there is gene therapy, right? Gene editing, it involves gene editing also, stem cell research, diagnostic, cancer immunotherapy, everything comes under the medical biotechnology. Then there is development of the monoclonal antibodies, then nanomedicine, which is very recent. Then personalized medicine, it is under the process, right? Personal medicine is right now the niche of the medical biotechnology research. Then for the agriculture biotechnology, which is called the green biotechnology, the genetic modification of the crops for pest resistance, higher yield and drought tolerance to basically make them resistant to the environmental obstacles. Then biopesticides, biofertilizers, plant tissue culture, animal breeding using the biotech tools. There are so many application in the agricultural field. Then comes to the industrial biotechnology, which is the white branch basically. So they uses the microorganisms and enzymes for production of some chemicals or biofuels and some biodegradable plastic also. Then there is fermentational technology that will improve the manufacturing efficiency and environmental sustainability. So lastly, the use of the biotechnology in this industrial biotechnology is on so many levels. Like we are dealing with microorganism, we are dealing with the fermentation technology. There is so much to go on, right? Then comes the environmental biotechnology. Here's the bioremediation of the pollutants using the microbes or the plants. You may have heard about it. The phytoremediation, in which we uses the plant to basically take off the beat the heavy metal from the ground. So basically plants are being used for the that means. So there is wastewater treatment, there is pollution control, restoration of the ecosystem, development of sustainable practices to protect the environment. Everything involves the biotechnology. So this is called environmental biotechnology. Then finally there is food biotechnology, which uses the microbes in food fermentation, like production of the basic table staples like yogurt, cheese, alcohol. Then there is development of the genetically modified food, which I was talking earlier, with improved nutrition and shelf life. We will be discussing each aspects in coming lectures. Then the functional food and probiotics, which are being developed under the biotechnology, which is food biotechnology. Now, how biotechnology works? So there are so many ways in which the biotechnology enhances the production or helps in these all fields. So one of that particular way is gene manipulation. What is gene manipulation? So gene manipulation means to deliberate modification or alteration of an organism's genetic material to change its characteristics or function. So genetic material, so that means we are basically dealing with either DNA or RNA, right? So this is the genetic material. So we will be on purpose modifying this particular genetic material to basically enhance or get the desired product. So you will be changing the characteristic or function of that particular organism, and it involves techniques which allows the scientists to either add, remove or modify specific genes within an organism's DNA. So that means you are going to manipulate the genetic imprint or genetic impression of that particular organism. So the concept of the gene manipulation is that it enables the control of the gene expression and protein production by altering the gene sequence. If you are aware of the central dogma, so you have DNA, then you have the messenger RNA, and finally you have the protein, right? So the protein is the main thing basically, which is the the information in DNA carries the carries the message all around from the messenger RNA, and then finally you get the protein. The functional unit is protein actually, right? So if you can change the gene expression, you can alter the protein production also, because this is connected to each other. So that's how the gene manipulation work. You will control the gene expression and the protein production by altering the main thing, that is the DNA or RNA. And it uses the biotechnological tools to make targeted changes for research. That means ऐसा नहीं है कि you will be changing the whole DNA. No, you will be picking some target and then you will be changing that thing only, right? So they can be for research, they can be therapeutic target, agriculture target, or industrial purposes. For example, a very basic example is if you want to kill cancer cells, right? So for that you may do the therapeutic targeted therapies. For example, we are dealing with the breast cancer, so there may be some kind of gene which is directly involved in the breast cancer, so you can target then that gene, and then you can have the therapy for the breast cancer. So that is the targeted approach for it. Now, what are the common techniques for gene manipulation? So first of all, there is recombinant DNA technology, very very common. This is also called the genetic engineering. So first of all, there is isolation of a gene of interest from the donor DNA. So that means there is a recipient, there is a donor. So from the donor you will isolate the gene of interest which you want. Then insertion of this gene into something called vector. A vector is something which will deliver the gene to the recipient. Often it is a plasmid, right? And introduction of a vector, then this vector is going to be introduced into the recipient, which is basically your host, and it will be producing that particular gene expression that you needed, right? So what happens over here, you can see clearly in the picture that there is the DNA of interest. Then this DNA of interest is being used, basically this is the DNA that you want into the host, right? So this is the bacteria. You have collected the plasmid from the bacteria. The DNA of interest and the plasmid digest with the something called the restriction enzyme, which is very very useful in genetic engineering. So then this restriction enzyme basically cuts the plasmid and the DNA of interest in the same way, so that they can basically attach with each other. Then there is one enzyme called DNA ligase, which helps with the attachment, or you can say the ligation of the DNA of interest with the plasmid. And it now this is not a plasmid DNA, this is the recombinant DNA. This recombinant DNA is introduced into the bacterial cell. They will divide, and this DNA will be multiplied as the bacteria divides. So recombinant DNA copied, and you will get your product as such. So this is the main idea. Next, there is something called the site directed mutagenesis. So it introduces a specific mutation at a predetermined site in the gene sequence. So you have the gene sequence, and in that gene sequence you have a particular, मान लीजिए this is the gene, you have this particular site. So there is a predetermined site in the gene sequence to study the gene function or improve protein. So you either can use this one to study the gene function or to improve the protein. So this is basically used for protein engineering and functional genomics. Basically to just check what is the function of that particular gene, right? So here you can see this, there is inserted gene of interest, just like the previous example. We have a plasmid, right? So we are starting with this now, right? So we have a plasmid which have a inserted gene of interest. Then you see there is strand separation, the primer with the desired mutated sequence. So there is your desired mutated sequence with basically for which you want to see whether its function or whether you want to improve the expression of that particular thing that you are going to be using, right? So there is strand separation, then particular a primer is used with the desired mutated sequence. Then there is strand synthesis by the DNA polymerase using that particular primer. And finally, the digest wild type DNA, transform bacteria and generate daughter cells. So there is wild type DNA and then there is mutated DNA. Mutated DNA, because you can see there are both green and orange. So in the mix you will have both. So you will also get the mutated DNA and wild type DNA, and you can see the difference in the expression also, right? So you can use this technique for functional checking, basically functional genomics, right? Next is something called gene knockout and gene knock-in. This is also very commonly used. So in gene knockout, it disables a target gene to study its function or remove the undesirable trait. So basically, मान लीजिए these are 1, 2, 3 genes. You will delete this gene and see what is the expression. So if particular protein will not form, so you'll get to know that protein used to be formed by this gene, right? And in gene knock-in insert, where you will do what? You will insert a gene, right? So in knock-in you will insert something, and in knockout you will basically disable a target or remove it entirely. So in knock-in, you insert or replace a gene at specific genome location using a homologous recombination or engineered nucleases. Like you can see in knockout, this is the non-homologous end joining technique. Basically, this is a very common thing which happens in the DNA. So they are using the indel creation, in which basically there is knockout, they are deleting the gene, and in knock-in they are going to be using the hybridization technique, homologous recombination. They will be inserting this particular sequence, right? So both things can be done. You can either delete the gene or you can insert the gene sequence based on whatever research you are doing. Then the genomic editing is done with engineered nucleases also. This is very common practice nowadays. So using nucleases, for example, like zinc finger nucleases and CRISPR-Cas9, which is very commonly being used these days. These basically cuts are made at a precise DNA location using these nucleases. What is nucleases? This is basically an enzyme, right, which breaks the or makes the cut at a very precise location in the DNA. So cellular repair mechanism then introduced the insertion, deletion or corrections, and CRISPR-Cas9 is basically being used for its simplicity, because you just need the enzyme and it will do the work, and it can work at multiple genes simultaneously. That is its advantage. So in this, here is this example. You see that this is the patient. So you get the somatic cell from the patient, and you also get the stem cells from this patient. Somatic cell is being reprogrammed and the induced pluripotent stem cells are being formed, right? So this is the laboratory work. Now what will happen? This is the normal cells of that patient. The one is the iPSCs, next is the HSCs, right? So normal जो आपके hematopoietic stem cells हैं, basically those are there. Now you can see in the gene editing, what they have done, they have used CRISPR-Cas9. So gene addition, gene disruption, gene correction, all these things can be done using CRISPR-Cas9. And then the corrected hematopoietic stem cells are being inserted into the patient, or the corrected induced pluripotent stem cells are going to be differentiated into hepatocyte like cells, and then they are transplanted into the mice, or they can be directly transplanted into the that particular patient. What we are doing here is, this is called the personalized medicines. Okay, this is the personalized approach. So basically you take the cells from that particular person, and you change the expression in those cells, and then they are being inserted into the same patient, but these cells are only going to be used in the same patient. So this is a personalized medicine approach, and this is very common these days, not being implemented entirely, but this is the focus of the research these days. Now finally, there is something called RNA interference, which you may have learned about in your molecular biology also. So there are two ways. There is small interference RNA. There is microRNAs, miRNAs, which you call. Both mechanism basically do what? They silence the gene. They silence the gene expression, right? Because from DNA there is mRNA, and mRNA expresses and you have the protein. If mRNA doesn't express, there will be no protein at all, right? So small RNA molecule like siRNA or miRNA, they silence gene expression post transcriptionally. This is very, very important, right? So in some diseases, if there is some kind of gene expression which is certain, that if this particular gene will express, the particular disease will occur, so you can target the gene for its silence for that gene. So you can cleave that mRNA, or you can just make that mRNA in such a form that it will not express. So these are used for functional studies as well, or therapeutic studies. So basically what happened, there is a particular protein complex that is called dicer in both cases, and you can see this is a double stranded RNA. Normally in our cases in eukaryote, there is no double stranded RNA, right? So this is like a message to the cell that something is wrong, right? If any double stranded RNA is there in the human body, if we say, so this is like a message, right? This is not normal, because usually the virus contains the double stranded RNA, we don't contain it, right? Then these RNAs are basically cut down from this dicer complex, and they have this imperfect match chain, and then there is something called risk complex, which basically cuts the both ends, and then both strands separate from each other. There is one called guide strand, and that particular strand will be binding to the target mRNA, right? So you produce the micro or small mRNA, and from there after the processing, there will be single stranded RNA which is going to bind to the target. So these targets are complementary to the guide strand of the small interference RNA or the miRNA. So that is the whole point. After it binds, the translation will be blocked, or either the mRNA will be cleaved, right? So that there is no purpose of the truncated protein will be formed even if the translation occurs. So this is the whole point. So you can see there are so many ways in which the gene manipulation can be done. So there is number one, recombinant DNA technology, then there is site directed mutation, there is gene knockout, gene knock-in, and gene editing by the engineered nucleases, and finally there is RNA interference. So what is the purpose of doing all this? So the applications are in so many fields, as I have told you earlier. First is the medicine, then there is agriculture, research, and industry. So if you see particularly, insulin is a very common thing which we need for diabetic patient, right? So earlier insulin used not to be available that easily, but after the genetic engineering has produced the humulin, right? First they produced the something called humulin, which is very similar to the insulin. Then the exact formulation has been made by the pharmaceutical, but this has been possible because of the biotechnology. And then we have the vaccines, gene therapies for genetic disorder. All these things are possible only because of the biotechnology. In agriculture, we can have the modified crops with desirable trait such as pest resistance, for example. There are so many varieties of rice which is high in the beta carotene, and so many other things, right? So like golden rice, and then you have other food products which has been enhanced you called them fortified food products, right? So you have or if you see the crops, some crops are especially there is bt cotton which is resistant to particular infection. So these traits have been inserted to those crops using the biotechnology. Then comes the research, which is the most important thing. You need to understand the gene function and the disease mechanism. So for that you need biotechnology. And finally, in industrial basis, you need engineering microbes for enzyme production, bioremediation, and so many other things. So this particular field has so many other applications. So I hope the essence of the biotechnology or animal biotechnology is clear to you. In further lectures, we will be continuing further topics of this particular section of the unit. If you have any question, please write it down in the comments. And if you understand something, please like the video and share it with your friends who can be benefited from the lectures. Subscribe if you are new and stay connected for upcoming lectures. Thank you so much for watching till here.